Okay, well, hello everyone. Welcome to ARC SEAM webinar series. My name is Nishar Hameed. I'm from Swinburne University, and I'll be moderating today's session. I hope you all are keeping well and safe wherever you are, and looking forward to coming out of the lockdowns. Now, before we start, I would like to acknowledge and pay our respect to the traditional owners of the land. We respectfully acknowledge the Wurundjeri people of the Kulin Nation, who are the traditional owners of the land on which Swinburne's Australian campuses are located in Melbourne East and Outer East, and pay our respect to their elders, past, present, and emerging. Now, in today's webinar, we have two distinguished speakers from two organizations. First, we have Dr. Helmut Thiessen from CSIRO, who will be presenting on surfaces for the detection and inactivation of pathogens. And then we have our own Professor Peter Kingshott from Swinburne presenting on colloidal crystals equals complex surface topography plus chemistry equals enhanced biocompatibility. Well, I don't know what it means, but I look forward to see how Peter is going to solve that equation. Now, I will introduce both speakers in the beginning. To briefly introduce speakers, Dr. Helmut Thiessen first. He obtained his PhD in chemistry from RWTH Aachen University in Germany, where he also started biomedical research at the Interdisciplinary Center for Clinical Research. He then joined CSIRO in 1998, where he now leads a team with more than 25 scientists that is focused on the topics of biomedical devices and regenerative medicine. Helmut has published more than 160 peer-reviewed journal publications and book chapters, and more than 10 patent families. He has won many prestigious awards, including CSIRO Medal for Research Achievement and the Newton Turner Award for Exceptional Senior Scientists. Now, coming to Peter Kingshott is the Professor of Surface Engineering in the Department of Chemistry and Biochemistry at Swinburne. He's also the Deputy Director of ARC SEAM. Peter completed his PhD at UNSW and CSIRO in 1999 and was a postdoc at CSIRO, University of Washington and RWTH Aachen. And then he became senior scientist at the Danish Polymer Center at Riso National Laboratory. He was an associate professor at Aarhus University at Denmark before joining Swinburne. Peter's research has a strong emphasis on controlling the interfacial interactions of biological materials with advanced material surfaces. Application areas include medical implants, infection control, tissue engineering, and regenerative medicine. Peter has published over 180 papers with an index of 50 and over 9,000 citations. Now, with that introduction, we will move into the presentation, and I also would like to highlight that we'll have an opportunity to have questions towards the end of the presentation. So please type your questions in the message box, and I'll be happy to pass them to the speakers towards the end. And before I hand over to the speakers, I also wanted to highlight SEAM webinar series. We have the next webinar coming on 5th of August by Professor Chris Byrne, our director. Is presenting on beautiful microstructures, case histories for designing the face of matter. So keep an eye on that particular date, look that date for the presentation. So with that, I'd like to hand over to Dr. Helmut Thiessen for his presentation. Over to you, Helmut. Thank you.
Thank you very much. That's great. Thank you so much for the very nice introduction and introducing a little bit about our background and our work. Of course, I would like to thank the organizers again for giving me the opportunity to speak here. And of course, CSIRO is a very active SEAM member, and so that's why I'm talking here today. I'm going to tell you a little bit about our work in the detection and inactivation of pathogens today. And of course, as you would imagine, in a SEAM presentation, it's all about surfaces. So it's really all all this important work that is happening in regards to biosensors and new technologies that inactivate and prevent pathogen transmission. At the end of the day, all of this is related to surfaces. Material surfaces and surface chemistry. And I'll go into a little bit of detail why that is so. Just as a reminder, CSIRO, of course, is our national research agency. It's actually the Commonwealth Scientific and Industrial Research Organization, if you spell it out. And our organization has delivered solutions for what the nation really needs, the environment, of course, business and our society for more than 100 years. We have more than 5,000 staff around the country, and we are by far Australia's largest patent holder. So that means there's a lot of emphasis on intellectual property. And of course, we, as you would expect, due to our role, we work together with a large number of industry partners every year. Building on that, of course, there's many success stories that I could talk about, related to the work that has happened at CSIRO. Ranging from the invention of Wi-Fi to, for example, something that has happened in our area, the development of extended wear contact lenses. While this is a good example to highlight here as a success story, it also is a good example for a biomedical device that actually easily can become infected. And you can see here that you're touching this medical device with your hand. Every time you put it in the eye, that's obviously in a non-sterile environment. So infection and the surface chemistry associated with this medical device is obviously of concern. I won't go into more detail here, but I will say that we are working on a large range of medical devices together with medtech companies here at CSIRO in the moment. And this really reaches from implantable medical devices, such as hip implants, to devices that are not indwelling, such as contact lenses, things like catheters. In the middle here, you see a urinary catheter, but also importantly, this ranges to biosensors and also devices that you use for in vitro work, such as cell culture tools. So we work on all of these biomedical devices, with our medtech industry partners. Now, I mentioned in the title that we work both, and in the previous slide that we work both on, that we work on a range of biomedical devices ranging from biosensors to, implantable medical devices. So let's first of all talk about these biosensors and the specific class of biosensors that we actually work on. Again, very importantly, we work essentially on surfaces and in particular on surface morphology, surface topography and surface chemistry to make these work. And so, we can obviously, if you think about electrochemical detection, why do we choose electrochemical detection over other methods? Well, simply because the detection capability is actually going quite low in terms of the detection range, so down to the femtomolar range. And it's a very simple experimental method and low cost. So in other words, this is something that can be scaled up and is and can be done in a in a label-free way. That's important as well. We are basically using different electrode materials to then establish functional groups on the surface. Again, that's surface chemistry. And then we covalently bind many different receptors on these surfaces. So in an example here, we are binding aptamers, for example, on surfaces to then specifically detect an analyte. In this work, which we do together with our close partners at Monash University, we particularly focus on porous silicon nanochannels, and that basically allows us to improve the sensitivity by multiple orders of magnitude again, and importantly, this very high selectivity and sensitivity can be achieved while maintaining this low cost and high scalability. And you can see some of these nanochannels, these porous silicon nanochannels here on the right-hand side, where these nanochannels have a thickness of 2 microns, while these pores are in the nanometer range, the pore size. Importantly, when you think about this, this issue of scalability, this can be actually done on a 4-inch wafer scale. And importantly, of course, when you think about translation, as we do a lot with our industry partners, then you need facilities to be able to do this. And I will particularly mention here the Melbourne Centre for Nanofabrication, where much of this work is actually being done. This is, of course, a joint venture between SIX University and also CSIRO, next to the synchrotron also here in Clayton. Importantly, this facility operates under ISO 9001, so under a quality management system. And so this allows this is very important for our industry partners and for maintaining the quality standards that are so important for this translation. Going back to the issue of the devices, of course, you all know that infection, is everywhere. It's something that has many different meanings in infection, and it can have many different sources. But importantly, all medical devices are associated with infection rates. So we have actually, in a review a few years ago, looked at the infection incidence over the lifetime of different medical devices. And just to pick one device where these are, the rate is really bad, is urinary catheters, whereas others, such as contact lenses, they can probably not be improved much further, as the infection incidence is only 0.1%. Anyway, there's a need for many of these medical devices to really be improved. Of course, all of this is with the backdrop that antimicrobial resistance is really a road train that is coming towards us that we need to address. And so CSIRO has recently established the antimicrobial resistance mission, which we are currently working on to address this problem with many of our partners. The medical devices and again, really surfaces at the end of the day, surface modifications, surface chemistry is expected to make a significant contribution in this fight against antimicrobial resistance. So my prediction would be that antimicrobial resistance following the current pandemic will come to the forefront and will completely replace our worries about viruses that we have in the moment, and people will focus much more on antimicrobial resistance over the next few years. Antimicrobial technologies that we can actually access and that we can combine in many cases include the prevention of attachment of microbes on surfaces, contact killing in response to immobilized antimicrobial agents, antimicrobial release from a surface, and of course contact killing in response to topography as well. You know, as I said many times before already, industrial translation is the key for us. And so when we think about coating processes or surface modification processes, they need to be highly effective. They need to be extremely reproducible. They need to be simple to integrate into the manufacturing process. And they ideally need to be one step and also cost effective, of course. Now, a very, very important point here is that our industry partners, you know, up until 1.5 years ago, would basically demand that we have a broad spectrum antimicrobial coatings or technologies, which basically meant coatings that can deal with gram-positive and gram-negative bacteria. Nowadays, that really has been broadened, that demand, and it really, industry is increasingly demanding pathogen prevention, actually meaning, prevention of bacterial attachment and infection, protection against fungi and really also against viruses. So, you know, it's become very, very broad, and you can imagine where this comes from. That comes from, of course, the awareness about viruses that really has been precipitated by the pandemic. Now, when we look at approaches that we feel most comfortable with, then we typically look at this picture here. We can either choose a grafting 2 approach where we have preformed polymers in solution that are coupled to a surface via a complementary functional groups, or that we have initiators on the surface and a monomer in solution, where we essentially have a grafting from process that ends up with the same picture. More recently, and over the last few years, we have exclusively used cross-linked coating approaches, simply because they can actually be turned into a one-step procedure, and they can essentially be translated much more easily into an actual commercial product. And so we have used cross-linked coatings consisting of multiple components, but also photo cross-linked coatings that basically exploit preformed polymers in solution that are simply deposited on a surface and then photo cross-linked. As one example that I will show you in a little bit more detail is an antimicrobial technology that we have published and of course also patented, where we have essentially a 3 component mixture, which is for example, a polyethylene glycol polymer chain with epoxy groups at the end. And then we have an amine cross-linker molecule, and we simply mix these together. And we can also mix these together with an antimicrobial drug. This antimicrobial drug also needs to have an amine functional group at least one end to be able to for us to cross-link this covalently into the coating. On the right-hand side, you see a spray coating apparatus that we use here in our facilities to get a very good control over the coating and coating thickness, for example, of these materials. When we use antimicrobial peptides for this purpose here as the antimicrobial drug, these have many different advantages. For example, they're less likely to develop resistance. They're very broad spectrum and have very high biocompatibility. But the disadvantage of these is that they have a very short half-life in vivo. We can address this by covalently immobilizing this on a surface, so that this is not these drugs are not cleared via the kidney so quickly, and so we can actually establish very effective coatings based on that. In particular, we have played around with an antimicrobial peptide called melamine with our partners at the University of New South Wales. And importantly, that is one of the few examples for molecules that have been demonstrated to be active when surface immobilized. So a lot of compounds have efficacy if they are if you use them in solution, but once you surface immobilize something, then in most cases you will not get activity anymore. So you have to be certain before you surface immobilize an antimicrobial compound that it will actually work on a surface. Otherwise, you might invest a lot of time into something that doesn't work. If we use these coatings that I mentioned before with one layer of defense, if you like, if we only use these coatings to provide to repel these bacteria on the surface, then if we look at protein absorption, that is, these coatings are extremely efficient at preventing protein absorption, as you see on the right-hand side. For an assay looking at adsorbed fibronectin. If we look at cell attachment, that is also very effectively prevented on these surfaces. And if we then go to mix in the the antimicrobial peptide as well, then we can see that this second layer of defense of having a repelling surface, a low fouling surface with this antimicrobial peptide, then that we get an additionally statistically significant benefit from this second layer of defense, and that's exactly what we want. We want to have this this combination of two layers of defense, so it works. Where is this important? Well, just to give you one example for an area where there's significant need, that's in the area of endotracheal tubes. Currently, you see at the top here an uncoated endotracheal tube that people, that is very highly relevant currently for COVID-19 patients who get onto a ventilator. That's uncoated, and so that's a perfect way of perfect entry for bacteria into your lung. And a lot of people die from secondary complications that are due to these devices. And so if we now apply these coatings, and if we prevent bacterial attachment and biofilm growth onto these devices, then we can make a real contribution. And here's an example for a study that we have done in this space. I also wanted to briefly mention that we have established not only these simple cross-linked coatings on surfaces that I mentioned before, which are still requiring an amine group on that surface of a substrate, but that we have also, together with our partners at NIMS, the National Institute for Material Science in Japan, a coating chemistry which relies basically on a polymer that has two components. First of all, a component, in particular the hydroxypropylacrylamide component, that is low fouling again, and secondly a component that allows covalent anchoring and cross-linking of these polymers. Basically, this benzophenone component that you see here falls apart under UV and produces radicals, which can then react with their environment. We have demonstrated that when we coat surfaces, different materials with this sort of polymer, that we can completely prevent cell attachment on surfaces, and of course we also can prevent bacterial adhesion on surfaces. I won't go into much more detail here, but I will say that we have very recently, in the last half year or so, we have incorporated peptides into these polymers by synthesizing peptide monomers. And as one example shown here, you can incorporate peptides that are targeting CD44, that are binding CD44. And so that gives you an opportunity, if you cast that onto a surface, if you cross-link that onto a surface, to specifically target different cell types. So only cell types that express CD44 on their surface will be captured on these surfaces, because they are completely non-biofouling, so cells can't attach on them unless this peptide, which is displayed on the surface, can interact with the CD44 on the cell surface. So only human mesenchymal stem cells were attaching on these surfaces, and this was, of course, as you would expect, concentration dependent. Now, obviously, this same concept can be used for introducing antimicrobial compounds, and Fei Huang, who is a PhD student at Swinburne University, is currently working on this topic, and so he's has special interest in particular in antibiotics that he attaches to these polymer chains, and also quorum sensors that he uses in this context. The last basically bit is just to say, when you think about antimicrobial technologies, we shouldn't overlook that, of course, topography also plays a big role in this context, and that these surface chemistry approaches can be certainly combined with surface topography effects. And so Elena Ivanova and others that you will be well aware of have done a lot of work in this area of mechanobactericidal activity, which was first discovered on insect wings. And essentially, there's nanotopographies that were discovered that create this effect. While a lot of people have looked at creating nanostructures that have, very high control over these structures, and that are, very require E-beams and other methods. Elena Ivanova and others have looked at, well, can we create random structures that can achieve the same effect? And turns out that is exactly what you can achieve. And so, for example, on titanium, simple hydrothermal treatment can achieve this bactericidal effect resulting from these nanostructures. And we've been involved in this work as well through a PhD student. And of course, importantly, these bactericidal activity can does not mind if these bugs are actually resistant against antibiotics or not. So this is definitely also a great strategy to deal with superbugs, if you like. But importantly, the combination of these mechanobactericidal approaches with surface chemistry approaches, that's the real opportunity going forward. So last not least, I would like to mention that we do have a facility here at CSIRO, the Biomedical Materials Translational Facility, where we can work with our academic and also industry partners on these surface technologies, this range of surface technologies, and also where we can do, in vitro experiments. And importantly, we can do all of this under quality management that we have established, ranging from ISO 9001 to the ability to do things under GLP. So that's important again for the translation. Obviously, there's huge market opportunities, not only in the biomedical area from this, but also in indoor air quality systems, hospital environments, food, textiles, and many others. So in conclusion, I think I think biomedical devices are, very important tools in the fight against pathogens. Pathogens really meaning not only bacteria nowadays, but also, the whole range of bacteria all the way to viruses. And of course, all of this is closely related to the fight against antimicrobial resistance, which is the next big topic, as I said before. And I think this is an exciting area and an area that is important for everyone. Before I close, I would just like to thank you all, and I would like to highlight that CSIRO is currently already and will be over the next few weeks advertise 20 positions in the biomedical area, and many of these positions will be ongoing science positions at many different levels. And so I would like to encourage all of you to have a look at the CSIRO jobs site, if that sounds interesting, because we would love to hear from you if this is something you're interested in. Thank you so much.
Thank you, Helmut, for the wonderful presentation and demonstrating and showcasing the research at CSIRO. Now, with the time pressing, we'll move to Peter, and we'll take all the questions towards the end of the webinar. Please post your questions in the message box there, and we'll discuss them towards the end. Now, I'll hand it over to Peter for his presentation. Thanks.
Thank you, Nishar. I'll just share my presentation. So first of all, I'd just like to thank the organisers for the opportunity to talk, and also Vesna for arranging this session. So my presentation today is talking specifically about a way of making new coatings, and the ultimate aim is to control biocompatibility, to enhance biocompatibility, and it combines a method based on colloidal crystals that simultaneously show great promise at controlling both the topography and the chemistry in one surface format. So let's have a little bit of background first. So if you take any biomaterial and you expose it to a biological environment, and Helmut gave a very good overview of the problems that exist in biomaterials. Of course, they do have lots of benefits in terms of improving quality of lives and even saving lives, but they are having inherent properties, and there's still a lot of research required to have an optimal biomaterial surface for a particular application. But it's the specific interactions of the environment that play a role. So, of course, mechanical properties of the material is important. The actual biological environment where cells and microbes are growing, including those that have soluble factors, drugs, small molecules, growth factors that enhance or inhibit attachment and growth of cells. Physical stimulation is important. If you come back to the material, you can actually manipulate the surface chemistry and the biological response by physical stimulation. The culture conditions are important, particularly the ones that you can control in vitro environments. But the aspects that we're interested in is particular control over the surface chemistry. So examples of some of the parts you would like to improve is enhancing cell attachment or preventing cell attachment by peptide grafting, but also preventing protein adsorption. If you have uncontrolled protein adsorption, then you don't really have any control over the cellular attachment events. And topography also plays a big role. It's been demonstrated quite significantly recently that the specific topographies really play a role in this so-called biocompatibility. So what I'm describing on this slide is essentially biocompatibility aspects that need in some form control over. And for us, as I say, chemistry and topography are the targets. So when it comes to control of chemistry and topography, we're aiming to have specific cues, and the level of complexity can vary from single factors through to complex factors that are essentially mimicking the extracellular matrix, so to speak, of a biological environment. So when it comes to single factors of a topography, you might have spikes, raised features, or holes. From a chemical point of view, you can have small molecules, polymers and peptides working individually to enhance a response. You can add a little bit more complexity here. You can have multiple factors which combine different types of topographies, or you can have those topographies having a uniform chemical signal, such as a uniform coating. But then you can introduce multiple signals in terms of the different types of molecules that either elicit a positive or a negative response. But ultimately, you want to control these events and have more specific interactions with the cells and and in a lot of cases the bacteria in the environment. But of course, you can get even more complex. So you can have surfaces that have multiple topographies, having multiple chemical entities that are distributed either randomly on the surface, or they're patterned in a controlled way where the morphology of the pattern is fabricated from some nano or microfabrication tools. When it comes to topography, there's simple ways of making different types of topographies. You can make porous materials through electrochemical etching. You can anodize surfaces to make nanotubes. Here's an example of titanium dioxide nanotubes. You can use anodization to make nanopillars, and you can use lithography, such as photolithography or E-beam lithography, to write structures in materials ranging from the micron scale down to a few nanometers. You can use simple techniques like electrospinning to create nanofibers made of polymers and hybrid materials that elicit positive biological responses. And you can make nanopillars using techniques like nano imprint lithography to transfer patterns into softer materials like polymers to elicit or potentially elicit some responses. So where do we fit into this concept of chemistry and topography? So one of the areas that I've been getting involved in the last few years is working with colloidal crystals. So colloidal crystals are combinations of particles that can be self-assembled on surfaces into different types of patterns. And colloidal crystals have been around for a long time, but recently it's been shown that they have the potential to be used, particularly in a medtech context. Now, there are many application areas, and we won't go through those. I mean, electronic devices, photonic devices come into play, and there's a whole range of methods to fabricate them, from simple self-assembly through to controlling the self-assembly process with electric-magnetic fields, etc., assembling at a air-water interface and transferring your substrate through that interface to create the crystals on a surface. And the degree of complexity can be changed. So here's an example of using single colloidal particles that are just spin coated onto a substrate, and the conditions of the solution, the properties of the solution, and the spin coating process allow for crystals of different morphology to be generated. And you can go even more complex, so you can add more than one particle to your solution, and you can generate colloidal crystals called, in this case, binary colloidal crystals, where you have a combination of small and large particle. The example here is showing, it's from Van Bladeren's group, who's one of the world leaders in colloidal crystals. He's demonstrating that inside of capillary tubes, you can actually control the crystal morphology by the size ratio and also the volume fraction of particles that you use. So there's a phase diagram that's been developed for forming crystals in three-dimensional structures. So when I was reading some of these papers about 10 years ago, I thought, okay, well, perhaps this concept can be applied to a biomaterial surface. So we started working with binary colloidal crystals, and what we demonstrated early on is that if you take low volume fractions of mixtures of particles of different size and different chemistry, and you just simply drop cast them onto a hydrophobic surface from water is the solvent, allow the solvent to evaporate, what you end up with is what is a crystal type of material that's just a solid deposited on the surface with a classical coffee ring type structure. Now, if you zoom in on this coffee ring structure, what you see in the middle is a very disordered layer of particles. They're randomly distributed. But if you actually perform some microscopy on the ring and zoom in, what you can see there is that you have some very highly ordered colloidal crystals, some binary colloidal crystals in this case. And what we were able to demonstrate is that this process of the crystal formation was independent of the chemistry of the particles and the size of the particles. So what you see here is an AFM picture of two different types of, or the same chemistry of particles, different sizes. You can actually change. You can have chemistries ranging from hydrophobic to charged to uncharged, etc. So this sort of opened up the possibility of creating a chemical pattern on a surface where you're presenting multiple chemistries to an environment like a biological environment. But the challenge was to try and obtain these structures over a fairly large surface area where you could test them in environments where bacteria and cells are present. So what we showed is that you can do that by controlling the evaporation of the solvent. So if you take your suspension of particles and drop cast them inside a rubber ring, and you control the volume and you control the concentration of particles, you end up being able to control the evaporation rates of the solvents to the point where crystallization takes place, and in a lot of cases you get these ordered crystals over the entire surface area within the rubber ring. So you can fabricate these assemblies over fairly large areas quite easily. And what we also demonstrated is that you can use a range of different particle sizes from a few nanometers up to a few micrometers with different materials, with materials having different surface chemistries. You can also induce order and have that as a complement to your ordered systems. But the nice thing is you could also deposit these on different substrates. So glass, silicon wafers were always used because of their model systems that we employ for characterization, but you could also deposit them on gold, different polymers, tissue culture plates, metals like titanium, for example. So here's just some of the early examples of the types of crystals you could form. These are the binary colloidal crystals forming the ordered structures. And what you see here is essentially a range of SEM pictures showing highly ordered BCCs with different combinations of particles of different size and different chemistry. So ranging from a few micrometers down to say 50 nanometers. You see these very large ordered structures. So this gives you the opportunity, if you're looking at a biointerface, to have multiple signals being presented to a biological environment on the one surface by simple self-assembly. So you pre-modify the particles, or you don't modify the particles, assemble them on a surface, and then you see what response they have to the target, which is either a cell culture environment or a bacterial culture, trying to understand how bacteria attach to surfaces with these types of structures, which is something that's not that widespread in the community. So here's just some pictures showing that you can, in fact, create these structures over fairly large areas. These are low magnification SEM images, and then zooming in to show the ordered structure. So it is possible to make them over fairly large areas. We did extend on this work to add more particles to the suspensions to see whether they could self-assemble into structures. So what you see here is some SEM pictures of colloidal crystals made from 3 or from 4 combinations of particles in the one suspension. And clearly what you can see here is you're getting some ordering within the larger scale ordering of the larger particles. So the subordering within the ordered structure. So it is possible to make combinations of crystals where you have up to 4 particles, all of different size, all of different chemistry, being presented to a surface. And this is quite exciting because you might envisage the ability to present or attach 4 different biological species to each of the particles and make a highly complex surface that can have some sort of positive response on, in our case, on cell culture, stem cell culture, etc. You could envisage having a surface that has, for example, peptides that elicit cell attachment, they cause cell attachment, but also having peptides that perhaps kill bacteria or prevent bacteria from attachment. So in a biological environment, this is aiming to elicit a positive response where you have tissue integration, but you're preventing infection. And if you can have a surface that has the complexity to be able to do those things on one type of surface, then you have an advantage. And that's the thought process in utilizing these types of systems. What we found moving on with this work is that there is a window of opportunity. Not every combination of particles will form nicely ordered assemblies. And we demonstrated that you need to have a particular surface charge and also a particular size ratio to fall inside the window where you get highly ordered structures. Outside of those properties, you can get crystals that are disordered, or you can have adsorption, so you have particles adsorbing to each other and then forming so-called raspberry-like particles on a surface. And these are also interesting surfaces because you can compare to ordered systems, and you can also make some quite complex materials just by simply getting particles to assemble together. So just to show you that structures that are disordered, here's some SEM pictures of different combinations showing a lack of order. So all of these have particles that are randomly distributed on the surface, which is good to compare to the systems that are ordered. And here's just some examples of particle combinations where you get adsorption of particles, for example, small particles to the large particles. You end up with some raspberry-like particles in some of the systems. And these might be interesting as well because you're introducing topography to, in this case, a large particle. You could envisage making particles that are raspberry-like, and then using them to assemble into structures where you have that hierarchical topography being presented to a biological environment. So we're taking these systems and then we're studying how they behave in a biological environment. So what we found out quite early on is that you actually have to stabilize them, and we use heat stabilization. So we take assemblies of particles. This is an example of polystyrene and PMMA and silica and PMMA of different size, 2 microns and 0.4 microns. We form the assemblies and then we gently heat them, and then they're stable enough for cell culture up to quite a few weeks. So they can be stabilized. And we initially started working with some simple cell types like fibroblasts and osteoblasts, but extended the work to stem cells, where the aim is to utilize the combination of chemistry and bioactive signals to elicit desirable differentiation of stem cells. We do a lot of characterization of these systems. Apart from SEM, we use AFM to zoom in a bit more and see the topographies, but also calculate the roughness. This is a combination of different particle sizes, and you can see you have control over the average roughness. You also have control over the wettability. So topography obviously affects the wettability, which is also something that affects biocompatibility. So we perform contact angle measurements. We also look at the chemistries with techniques like X-ray photoelectron spectroscopy and time-of-flight secondary ion mass spectrometry, just to make sure that we have the chemistries we're interested in on the surfaces. Let's have a look at how some of these surfaces behave in cell culture. So what we can see here is that fibroblasts attaching to colloidal crystals made up of polystyrene, 2 sizes. We can see quite clearly that the fibroblasts attach and grow, but they stay in a rounded morphology when you compare that to the standard tissue culture polystyrene. So you consider that tissue culture polystyrene is made up of the same material as these colloidal crystals. The only thing that's different here is the topography, in essence. So the topography here is allowing control over the cell morphology, which is a first start in controlling the cell behavior. If you can control the cell morphology, if it's a stem cell, for example, you have the potential then to control the gene expression, which then controls how it differentiates. So having the ability to do that is, in the first instance, what we're able to achieve here. And if you zoom in on some of these fibroblasts on the surface, you can actually see the colloidal crystals underneath because they get stained as well. One of the interesting things we found out early on is if you have a subtle change in the chemistry of the particles. So what we have here is exchanging polystyrene for silica particles and depositing them on the surface, and then looking at cell culture, you can see that just a simple change of a particle in the crystal changes the morphology of the cell. So on the silica surface, you have more rounded up cells, whereas on the polystyrene PMMA surface, the cells start to elongate and they send out these so-called filopodia. So there's a complete difference in behavior of the same cell type on a very subtly different surface in this respect. We've also extended this work to look at stem cells, and look, in this case mesenchymal stem cells, and we can show, or we have shown, that the crystals, some of the crystals do in fact control the morphology of the stem cells and control the way they differentiate. So this is just showing some nice images of the cells as they grow over time, and then calculating the number of cells, or quantifying how many cells are on a surface, and then looking at the average cell area, the area per cell, which is a measure of the morphology. So we can control how stem cells behave on the surfaces as well. And we have a few papers showing how they can be, as I say, differentiated, but also we've studied converting somatic cells into stem cells using these surfaces as well. They do have nice control over cell behavior, and these are all unmodified particles. So what we have extended this to is to actually now start to introduce some biological signals to the particles themselves and forming the assemblies. What we also want to prevent is the non-specific absorption of protein. So coating the particles with non-fouling polymer brushes, but then having the biological signals attached to the end of the polymers to elicit that response. So here's just a schematic of one of the systems we've used. We've taken silica particles, we've modified it with the polyethylene glycol, or we've taken the same particles and then modified it with a chemistry that can be then used to attach a peptide which elicits a very positive response in terms of cell attachment. And then we've made these particles, modified them, and then assembled them into crystals. As I say, we utilize XPS a lot, so this is some XPS data showing particles after modification, and you can clearly see the differences in the surface chemistry. In the middle here, you have particles that are modified with polyethylene glycol. You can clearly see the glycol from the backbone, or the ether component from the backbone of the polymer. This is with a peptide. You can see this amide component, which is indicative of the peptide backbone. So the XPS confirms that we have successfully modified the particles. We perform SEM again to see whether we form the assemblies, and generally we get very good ordered structures. We've also done, in this case, some cell attachment. This is osteoblast cell attachment, and what we can see is that the crystals that have the peptides elicit far more cell attachment and growth rates compared to crystals that have the polyethylene glycol, preventing adsorption or attachment on one particle, and also compared to unmodified particles, where you get good attachment but not as good as when you've modified the particles with the peptide before assembly. And you can quantify the images. This is a busy figure, but it's just showing that some combinations you get higher cell density, particularly those with the peptides, compared to the gold standard, which is the tissue culture polystyrene. So the combination here of the bioactive signal plus the topography is actually giving you enhanced cell attachment and biological responses. I'm sort of running out of time a little bit, but I just wanted to mention that we're making more complex modified particles for forming these crystals. We're using different types of peptides, some that are more bioactive, for example, using a cyclical RGD, which has been shown to be more active than the linear RGD. So we're making this, and we're making particles that have combinations of polymer brushes plus the peptides, and they are showing a lot more promise than the previous example I showed, where you just had a single peptide on one particle and a single polymer on another particle. This is just showing some SEM pictures of the systems, showing that we can form the assemblies with different combinations of particles. And this is showing some cell density and cell area results for cells attaching to different combinations compared to the tissue culture. So clearly we're seeing enhanced cell growth and cell areas when we are modifying the particles prior to their assembly. And these systems with, in this case, the cyclical RGD are certainly behaving much more better than a linear RGD. This is demonstrating some mesenchymal stem cell work where we're showing that there is a positive response for these crystals on not only stem cell attachment in terms of numbers, but also the way the cells are behaving in terms of their morphology. And also when you're taking away some of the proteins that are present, you're using serum-free conditions, the surface itself is actually giving you a positive response compared to the unmodified surfaces. So having these peptides and the topographies presented to the cells is allowing us to do cell culture in serum-free conditions, which eliminates the problems associated with all the proteins in the serum during the culture. Lastly, on the stem cell side, we've looked at stem cell differentiation, and the take-home message is that the combination of polymer and peptide on top of a colloidal crystal is giving us greater differentiation of MSCs into osteoblasts. So we're having greater creation of osteogenic conditions, and this is studied with looking at the activity of alkaline phosphatase and also the deposition of calcium. So the assays are demonstrating that without these peptides and without preventing the serum protein absorption, perhaps you couldn't get there as efficiently as with the bioactive species, and also, as I say, preventing the non-specific absorption. I'll quickly finish up by saying that we're also looking at bacterial interactions with some of these colloidal crystals. So we are looking at how bacteria attach to unmodified crystals. We're also combining crystals with some thin film deposition, such as plasma polymerization, to create patterns. You can actually make chemical patterns because the particles act as a mask, and once you've deposited a plasma polymer, for example, or a gold layer, you can actually remove the particles, and then you have a chemical pattern that you can then functionalize. So this is an example of functionalizing the regions where there is a plasma polymer with polyethylene glycol, and then studying bacterial attachment, and you can actually see that you can selectively, or you can get the bacteria to selectively attach to the unpatterned or the uncoated region on your surface. We're also combining some of these systems with antimicrobial peptides and also some other biomolecules to enhance that sort of attachment, but also repulsion of bacteria. And one example is the use of just colloidal crystals by themselves, and then depositing a layer of highly adhesive plasma polymer on the top. So plasma polymer called allylamine is very adhesive to bacterial cells. But when you deposit that highly adhesive coating on top of colloidal crystals, you see a quite different response. What we're actually seeing in the case of Pseudomonas is that the bacteria seem to get trapped inside of the colloidal crystals, where the smaller particles are appearing within the crystal. Even in the presence of a highly adhesive coating, the outside surface, or the large particle, is non-adhesive towards bacteria. And you can quantify the attachment on those surfaces, and what we see is that all the colloidal crystals that have an adhesive coating do repel the bacteria in terms of the final number of bacteria, but they seem to selectively adhere the bacteria into certain regions of the crystal, which actually opens up a lot of possibilities in terms of controlling how biofilms form. If you can, for example, selectively attract a good bacteria within a biofilm, then potentially you can prevent infectious bacteria from being present and potentially prevent infection. And lastly, I just wanted to mention that we are also modifying the particles that form these crystals with antimicrobial peptides, and we're actually showing that these surfaces are quite effective against bacteria. They're still able to kill the bacteria. This is some live dead staining showing that an antimicrobial peptide that's attached to a surface has a greater killing rate compared to when you just physically absorb the peptide to the surface compared to the control surfaces. So if you're able to combine the topography with an antimicrobial peptide or some other antimicrobial agent, you have a surface that's even more effective against bacteria. Okay, I've gone quite long in time, so just to conclude, we are using colloidal crystals in many ways to create surfaces that are being used in biological environments. That is either trying to enhance cell attachment, cell differentiation, and potentially tissue integration, but also repel and kill bacteria. And we're also using these crystals in novel ways to create different types of surfaces, which I haven't talked about today. So you can actually use them to create highly complex surfaces when they're used as masks or as catalysts for depositing thin films, where you can selectively grow other materials from the different types of particles. So with that, I'd like to lastly acknowledge a lot of people from CSIRO, from Swinburne, collaborators at RMIT in Sydney, back in Denmark, also some international collaborators in Washington, CAS in China. We have some very good collaborations in two of the IITs, Madras and Hyderabad in India, also Nanyang and also the Baker here in Melbourne, and also some of the funding sources for the work. And also thank you for listening to the presentation, and apologies for taking a bit of extra time.
Thank you, Peter. Thanks for the great presentation on colloidal crystals there. So we have a number of questions I can see there, which is great. Because of the time, I'll probably touch base a couple of questions for both. So there's a question for Helmut from Paul. Is there any evidence that bacteria can adapt to topological antibacterial surfaces? So Helmut, if you can address on that.
That's a great question. I think you know, that's a question I might have asked myself. It's actually you know, the mechanism by which the bactericidal activity works, you know, through the surface topography, acts very quickly. So you know, the bacteria that settle on that surface, they die very quickly. So therefore, bacteria don't have the opportunity to you know, to adapt to that mechanism. You know, that don't have a long time. You know, if you think about bacteria that are being exposed to you know, antibiotics at a very low level, and that gives them a lot of time to adapt to this mechanism. So therefore, you know, the answer is essentially, you know, the expectation is that this mechanism will be, you know, will be with us, will be effective for a long time, because bacteria, you know, are not giving the opportunity to develop a counter mechanism. Eventually, this will happen, but it'll take a long time.
Thanks, Helmut. There's a couple of questions from Thomas again to Helmut, because it was posted earlier. I'll probably go to the second question, which would be of interest to many. So how easy is it for yourself or the group to spin out some of the biomedical technology at CSIRO into a startup or companies? Is it typically achieved by existing partners, or can you do it by yourself as researchers within CSIRO?
Yeah, so CSIRO itself, you know, will only on rare occasions spin out a company. So that's not a typical model. It is much more likely that we will develop technology with our industry partners in projects that we have together, or that we develop technologies in-house or with our academic partners, which then, you know, will publicize, and where, you know, we can perhaps attract an industry partner that we can help to grow. So that's probably the much more likely model.
Thank you. So that's a question for Peter from Thomas Patterson. Thanks for the talk. Beautiful images. What is the maximum thickness that can be achieved with the deposited colloidal crystal films? Is it simply limited to the amount of material deposited, also in the order maintained throughout the depth of the films?
Thanks, Thomas, for your question. The way we control the thickness is simply the way we get a single layer. First of all, is to calculate how many particles we need to make up that single layer, and then if we want to make multiple layers, then we just double up, triple up, etc. What is the maximum thickness? We've achieved layers up to 7 large particles thick, if you can use that as a term. And they seem to generally, the ordered systems seem to remain ordered throughout the bulk of that layer. We haven't gone beyond that. Whether it's possible, I'm not sure, but I think it might be possible. It will also depend on the particle sizes that you're using, but also the area that you're hoping to cover. So as I say, beyond 7 large particles that are 5 microns in size, we haven't gone beyond that. So that's sort of the ballpark for us, at least.
All right, just what probably take this as the last question because of the time. This is a question from Nisa. Really good images. Great to see the periodicity in those images. Do you have any strategies to maintain the order of these materials? I think it's probably referring to long range order for a large area.
I think it does depend on the, well, it's a number of factors. In terms of the large range area, the method we use, if you can control the evaporation rate over larger areas than we use, yes, I think you can make long range orders, ordered structures. The longest range we've made is, or the largest area we've made is about 3 centimeters squared. But it also depends very much on the surface chemistry, and colloid and surface chemistry, people will know that colloidal stability is important. So if you can stabilize the colloids using different chemistries, whether it's charge repulsion or utilizing steric stabilization with brushes, then in principle you have a way of actually creating long range order that way. What is known in the field of crystallization is once the solvent starts to evaporate, then there is a critical volume fraction that is, when the critical volume fraction is reached, I think it's about 0.45 of solid to solvent, then entropy starts to take over and you get spontaneous crystallization. So if you can keep particles repelling each other up until that point, then I think you will end up with a long-range ordered system. If they start to aggregate and collapse out of solution before then you've lost your chance. So I hope that answers your question, Nisa.
Thanks, Peter. I think because we've gone over the time, so we'll probably stop it there. But please note that you can ask questions. You can email seam@swin.edu.au, or you can email both of the presenters with your questions. And also, I note that this presentation will be available as a recording later down, so you can watch it. So with that, I would like to thank both Peter and Helmut for their wonderful presentation and taking time to be here, and also all of you to attend this session and spending your time here. And also thanking Vesna and the AV team for organizing everything beautifully. And just finally, stay safe, and I'll be in touch with you soon. Thank you.
Thank you. Bye-bye.